Welcome back. In this episode, we'll be covering cellular respiration, oxidative phosphorylation, and photosynthesis. We'll start this show off with cellular respiration. The first step in cellular respiration is glycolysis. Glycolysis breaks down glucose and forms pyruvate with the production of two molecules of ATP. The pyruvate end product of glycolysis can be used in either anaerobic respiration if no oxygen is available or aerobic respiration if oxygen is available. The next part is pyruvate oxidation. Pyruvate oxidation is essentially the conversion of pyruvate molecules to carbon dioxide acetyl coenzyme A and NADH. Next is the Krebs cycle, also known as the citric acid cycle. The Krebs cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetyl-CoA, derived from carbohydrates, fats, proteins, into carbon dioxide and chemical energy in the form of ATP. That was the three main parts of cellular respiration, but next we're moving on with oxidative phosphorylation. The first step in oxidative phosphorylation is the electron transport chain. An electron transport chain is a series of compounds that transfer electrons from electron donors to electron acceptors and couples this with the transfer of protons across membrane to create H2O. The next step in this process is chemiosmosis. Chemiosmosis is the movement of ions across a semi-permeable membrane down their electron chemical gradient. More specifically, it relates to the generation of ATP by the movement of hydrogen ions across a membrane during cellular respiration or photosynthesis. Lastly but not least is photosynthesis itself. The first step in this process is caused by a reaction dependent on light. The light-dependent reaction in photosynthesis is the first major part in the process, in which light energy is initially converted into chemical energy as ATP and NADPH. This takes place across the chloroplast thylakoid membrane between the chloroplast stroma and the thylakoid space. Secondly, we have the Calvin cycle. The Calvin cycle refers to light dependent reactions in photosynthesis. That takes place in three key steps. Although the Calvin cycle is not directly dependent on light, it is indirectly dependent on light since the necessary energy carriers, ATP and NADPH, are products of the light-dependent reactions. The first step of the Calvin cycle is carbon fixation, and that's when a CO2 molecule combines with 5 carbon acceptor molecules, ribulose, biphosphate. This step makes a 6-carbon compound that splits into 2 molecules of 3 carbon compounds, 3 phosphoglyceric acids, or 3 PGA. This reaction is catalyzed by the enzyme RUPB carbolase oxygenase, or Rubisco. The second part is reduction. In the second stage, ATP and NADPH are used to convert the 3 PGA molecules into molecules of a 3-carbon sugar, G3P. This stage sets its name because NADPH donates electrons to or reduces a 3-carbon intermediate to make G3P. Step 3 is regeneration. Some G3P molecules go to make glucose, while others must be recycled to regenerate the RUBP acceptor. Regeneration requires ATP and involves a complex network of reactions